So this will be a little talk about our efforts to get whatever we do on the veterinary side to try to bring this to human. I guess the history of surgery, modern surgery, I would say, has been based on taking human experience, especially if we talk about orthopedics and trauma, and bringing this to the veterinary side. And when we started with Kyon in '99, and then after the first few years, we thought we have something useful. Maybe this would work for humans as well. And we were rather optimistic. 5 of us set up a company called Scion Orthopedics. Steve and myself are 2 of these 5 people. And we thought we're going to move basically with the same model that worked so well on the veterinary side. And there are some good reasons here to try this. Liability is still in veterinary side is much lower than in human. Human companies are constantly under threat of being basically taken out of business by all sorts of problems that come up, and then they get blamed and sued for that. Regulatory restrictions hardly exist. FDA does not regulate veterinary devices. In Europe, there's nothing. The only major market is Japan, where distribution of veterinary devices is controlled pretty much the same way as on the human side. The good thing about it is, as much as we can tell, surgery in veterinary side is much more difficult. What you have to do compared to humans is actually harder. And if it works in dogs, it's going to work in people. That's pretty much so. And then the use that animals put on whatever we do is much more demanding than what patients do, certainly for orthopedics. And I'd say for the fracture as well, you can't tell the dog not to walk for 6 weeks. I think on the veterinary side, at least in the last 20, 30 years, financially, these little companies like Kyon actually could survive doing whatever we've been doing, and that way contribute. So if you look at the projects that we currently have in Kyon that we think we can take to human, it is certainly the total hip replacement. It is possibly the partial, the cupless, maybe this Acetabular Hemi arthroplasty. And then the ALPS, the plating system, is a very peculiar example of this because it went from human to veterinary to now 2nd generation, and we look to bring it back to human. The shoulder replacement in dogs is hard to actually find good use for, but we have something that on human side might be very useful. And then, of course, the Ruby, the Arthrex, is the leader there, and they don't have anything that really works pretty good. And then there's the Patellar groove replacement. So we think all these things can be taken. Now, if you look at this, between 93 and 2019, there are about 18,000 hips. And we are at generation 6. As Aldo said, it's a completely new system for total hip replacement. With Scion, between 2003, so a few years later we started, 10 years later, we have in these years only 20 cases done, and it's still generation 1. It is, in a way, obsolete. At the time we started with Scion, Kyon was in generation 4, and there were some very good exchanges between Scion and Kyon. Whatever we developed on one side, we brought it back and forth between the 2 projects, I would call them, rather than, say, companies. Double shell metal backing was directly product of Scion efforts and demand that we had from that side, as well as the fossa cup, the reduced wear on the cup. That was done within Scion and then taken into Kyon. Steve did the stress analysis. You see here factor 2 reduction in stress. And then lubrication actually was tested by Nick Bishop at his lab in Hamburg. Factor 2 reduced friction, which resulted altogether in the factor 3 reduction of wear. And then we got into this clinical study with 15 patients, 5-year follow-up, which has been finished now. Everything looks as expected from the veterinary side, from dogs. in these patients, everything is sound. There's no displacement of the stem. There's no loss of bone anywhere on the cup or on the femoral side. So here's the summary of this. In 26 years, with zero invested, no capital investment if Kyon, 18,000 patients, and this hip created basically sustainable business. That's what Kyon was based on. And then on the human side, in 16 years, with $8 million investment, a good part of that comes from Kyon in the last 6 years. Without Kyon, this thing would have died. 20 patients, and calculate that. Well, I should have corrected 25 patients, but this is on my memory stick, Steve. I apologize. We do have 25 patients. So per patient, it's $320,000. And we still do not have approval. We cannot put this anywhere in any country that has regulatory demand. It is very disappointing. And meanwhile, here with Generation 6 and moving on, we're a good decade ahead of whatever is done in human, not only on Scion side, but on any other prosthesis, as much as we can judge. So the cupless is a huge challenge, whether this would ever work. We have 300 dogs here, 8 years of experience. On the human side, this might work with reaming acetabulum, which is how we want to do this. The range of motion and everything, this is working really good. You have a large head, thin neck. But we have a little bit of hope here to avoid this huge regulatory burden. If you try to register this as a regular device, either in Europe or here, it would take many, many years and millions. But as a Hemi prosthesis for neck fracture, it might be easier to get through. So without saying that we're going to ream, we're not going to ream acetabulum, we'll just put it in there. The bipolar is another possibility here, and then maybe as a custom device. For custom devices, you do not need FDA approval or CE in Europe. You need a prescription from the surgeon to do this. Now here's this maybe large disaster in orthopedics in the last decades. The hip resurfacing arthroplasty was a huge hit. Everyone wanted to do this some years ago. It started in 70s with Wagner in Germany and Amstutz here in the States. That was the first effort at doing just resurfacing rather than putting a stem on the femoral side and all that. And at that time, they used polyethylene on the acetabular side, and it did not work. It basically was abundant. In 80s was the end of that. And then there was this period where that was kind of forgotten, and then the next thing comes out. And this is McMinn, who came with Birmingham model with Smith and Nephew. He did himself about 3,000 of these. It was a huge hit. You look at the publications every year, kind of exponential growth. Everything's looking great until it didn't look good. And it's pretty much out now. Why was that? Because of metallosis. And then there was a bad name given to this tissue response, tumor-like tissue. And once that was out there, no one wanted to put one of those in. In some countries in Europe, it's basically outlawed. But it still survives. Maybe 20% of the numbers that were being used at the peak. This is what it looks like, a large head, which is cemented onto the femoral side, reaming out the femoral head to that shape, and then a thin cup for the acetabulum, usually cementless. Now, once you press that cup in there, a little deformation, the tolerances are very tight, there's friction in there, there's a lot of metal debris coming out, and these things turn into a disaster. Except, as you might have heard recently, people like Andy Murray, who wants to come back and be number 1 or 2 or 3 in tennis again. He just got one of these after dropping out of Australian Open a couple of months ago. So still those young patients are trying to get this thing so they can go back to full physical activity. There are a few of them with these implants. So the question is, can we come in and with a third wave of these what's going on here? Well, you can't do less than one implant. If you're going to do something, there's going to be just one piece. That's the end. And I think this is ultimate simplicity on that side. And if it's going to work, it's got to be ADLC or something like that, carbon coated for friction and wear. And we just do a touch on the femoral head rather than reaming the way you do it for Birmingham. Birmingham's still the leader, with about 20% of the peak use of that. Other companies gave up. And J&J is maybe facing extinction with the Pew, because this is the largest suit in history. It's going to be tens of billions of dollars that they may have to pay. That's going to drag on. But they may find a way out of this. It's 86,000 of these got implanted. Not all resurfacing, different types, but metal and metal, including resurfacing, and that has been recalled. So if we try this third time around, I think you've seen all of this now, and this is the picture of the dog one. And we just put the cup in there, we shape the head a little bit, put it in. Is that going to possibly work and give us a third chance at this? The advantage that we don't have on the veterinary side here is that on the human side, with Birmingham hip, the surgical approach was taught to and accepted by many, many surgeons, certainly hundreds, maybe thousands. So the approach is there. People know how to put it in. By doing this, you don't have the usual risks that you have by going into the femur, reaming out the femur, embolism and fissures, fractures, and all of this. All of that is gone. We don't have any of those risks. You can probably save 15 to 20 minutes of surgical time, which is definitely of interest. Implant cost, maybe more than a half reduction in the cost of the implant, possible cost. And then instrumentation is almost trivial now with this. So this next one is this long story of PC fix to ALPS, ALPS 2, and back to human, we hope so. The story starts in 1986. I kind of got under a door in the place where our workshop kept material. We had two people making prototypes in the institute, and they kept everything under lock. But I managed to kind of squeeze in, steal a piece of aluminum, and make one of these plates. By the morning, I had one of these with the holes and conical holes and everything. And I showed this to Stefan Perren, Fritz Straumann, Hans-Jörg Wies, a few of these people that were supposed to be really interested, and I would say they were. But this is between 1986. In the official report, sort of official by Kevin Newman, I think this was for 50 years of AO Foundation, they said 93, so there's some delay there. That started much earlier. I kind of like this slide. They couldn't figure out where I came from. But it was Stefan Perren and me doing this for a good 10 years. If I say $10 million invested in research on animals, that's not counting every penny. There must have been a lot more money spent on this. But then there was this crucial decision to be made. Are we going to replace DCP with LC-DCP or PC fix? And that made its way in. I tell the story in private. I will not tell it in public here how this went. But it was basically a bad decision, which was corrected later, a few years later, by AO demanding of the manufacturers to make this PC fix. And once AO demands something like that, the companies are supposed to respond. And so they did. Stratec in Switzerland, one of the three, put this thing in the catalog with the prices, but they never made any. That was the response. Here it is for you. And by the way, when Pierre Montavon tried to buy some of these to keep doing this in dogs, he used it in 60 dogs, the price of one screw now was $65. Not $6.50, but $65. The old ones were $6, and now they were $65. So the only thing that was right about that price list in the catalog were the prices. The plates were now hundreds of dollars instead of tens, and the screws were now 50, 60 instead of 5, 6. So there was something that happened there, but this plate never made it out. And the message, which I think is really the saddest story of this is from Emmanuel Gauthier, who spent years in Davos doing beautiful work. The message, keep it perfused, and if there is a movie that fits this, it's Lost in Translation. It was translated and locked the screws. So everyone has locked screws in every plating system today in the world, but the perfusion is not maintained. There is no system out there other than ALPS that maintains the perfusion of the periost and endosteum if you use short screws. So this is a message which is really wrong, but it's been perpetuated now for 20 plus years. That what we have, what you have today available from Synthes companies, for example, does the trick. It doesn't. There's nothing out there that does it. So why we did ALPS? I did ALPS because Foundation would not let me have, or should I say Synthes, let me have the license to do this for a veterinary side. And so in 2007, at our first symposium here, at this exact location here, we brought the first ALPS materials. And we sold a lot of that. That was the largest sale in a single day we have, I think, in the history of Kyon, mostly due to Japanese. All of you who came here in 2007 took this material back with you. And I think in these 10 years, or whatever it is now, 12 years, I'm sorry, all of this has been proven in clinical practice, whatever was expected, proven in animal experiments, proven in clinical trial, all that kind of came through with ALPS plates and all the details of development, different sizes and all of this. And the infection, this paper was published, took a long time until it was finally published, was presented by Dr. Yamaguchi at VOS. The resident of him presented it at the Symposium here in 2013, finally got published. And if you just look at this, 1 out of 271 clean cases not known to be infected got infected. This is available, it's online. And then personal communication, so this is not tight, you can't publish this, but these are numbers that I believe. We are looking at 0.3% infection out of about 700 cases here from 4 surgeons in Japan. I would say if you have if you have more than 1% infection, you should look carefully at what's going on. If you use ALPS, if you use TPLO, if you got 5% infection, you need to take care of the clinic. If you're at about 1, you're okay. Because if you take every possible measure to not get your patient infected, such as is done with the fancy hospitals where they do hips, laminar flows, scaphanders on all personnel, antibiotic in the bone cement, if you use that, whatever, you're still at 0.5%. You're not going to have 0, doesn't matter what. But you should not have 5%. So this is ALPS, and ALPS is going to turn into ALPS age. I think at least the visual thing is there still. I think these are pretty pictures. We have a beautiful photography done by a professional photographer in Zurich, whatever we do here. And this is a nice example of the plate. Nate just told us he had one of these put on his fractured bone. And the price was it 8,000? I don't see you, Nate, here. 8,000 was for the plate and the screws, wasn't it? Yeah. Yeah, but one of these plates with a few screws, $8,000 was the negotiated price. The asking was 15, wasn't it? All right, so this is absolute disaster what's going on. I can tell you that in 1986, when I brought that aluminum prototype to the AO people, technical committee people, they said, this is great. One size screw, you can just have a handful of screws. Those are going to be cheaper than what we make now, and that plate is cheaper to make than the standard one. That's going to be really good. It's going to cost $20 for the plate and $2 per screw. And you look what happens with our insurance systems. Europe is a little better, but not a lot. So here's the hip. This is the fastest growing joint replacement in the human side. Knees and hips are about where they are. But shoulder is still growing, and mostly with the reverse type. And this reverse type that we have now ready since about last summer, I think we showed it at ECVS. So we have implants, we have instruments. If anyone wants to do this, you're welcome. This is our Acetabular cup here. And this screw is the only thing that's different from the hip system. We use the ceramic heads here. It just seems that shoulders are not such a big problem in dogs. Suture anchors that Arthrex and other companies sell are absolute disaster, but they sell a lot of it, both to the vets and to the human. We have about 300 cases. There's nothing like that out there. Sports injuries, what those people, those patients get is just no good. It's mostly repair, maybe augmentation for other purposes, like including ACL. Arthrex sells things like that for augmented repair of ACL, not for replacement. And then there's PGR. We have about 3,000 cases here, as you heard this morning. What human patients get is this. Here's a bad one here, patella all worn out here. They get typically a cobalt chromium implant, cementless or cemented more commonly, with a little polyethylene plug stuck cemented into the patella. This is available. It's not as good as what we put in dogs. Maybe we can bring this out as a custom. And so I'll just take 1 or 2 minutes here. This is the ultimate journey that I've been on, and especially in the last years, we really think we're going to take this to veterinary oncology and possibly bring it to humans. This is another effort that is basically financed in the last 12 years or so out of Kyon. And Goran, who is here someplace, I think, or there, and myself are on this thing now well over 15 years. We have one publication, not a lot, but it did come out in 2013. It was done, the work was done here at Beth Israel by James Wells, who was a resident of Chris Evans, a friend of mine here. He is now at Mayo, and James did fantastic work on this. This is the best in vitro study done with arginine depletion, as what it became kind of known today. Dog cell lines respond, cancer lines respond very well to arginine depletion. He did 5 lymphoma, leukemia lines, and 7 osteosarcoma. And with arginine depletion plus arginase, essentially, for success of this, there were no surviving cell lines on the lymphoma side past 3 days. And actually, in 1 or 2 days, they were dead. If you look at these curves, that's just to illustrate this. Osteosarcoma took a little longer, 3 to 9 days of those 7 lines here. So what else is there? Well, arginase is much better than asparaginase. Asparaginase is a standard treatment for, as you know, for all these lymphomas in dogs. Thousands of dogs get asparaginase plus chemo. Now, this is what we've been after for 20 years now. At the end of this chromogram here is arginine, and you want to see 0. And we had 0 in about 2000 thereabout, and then we couldn't get it. All these years, until about a year ago now, we could not replicate the experiment we did in dogs. We've done a lot of dogs, experimental and few clinical. We couldn't get it down to 0. Where it has to be, that's not the only condition. But if you don't get this, you're not going to manage. And we think we know why now. It sounds a bit like what Keita showed you yesterday. What we do here, we take pig liver, we process it in reasonable ways, and with this, with hundreds of molecules in there, including arginase, we get that to 0. If you just take arginase pure, it will not do that. So we need this red juice here to make it work. We have some ideas why it works, but it doesn't really matter as long as it works. We're happy with this. So we hope in the next months and maybe a couple of years, we're going to bring this. We have a nice name for it, Hepaticos, registered here in the States. We think we can bring this to oncology on the veterinary side. what do we hope for? Well, we should get some lymphomas that we can cure. Not only help them live another few months, but actually get them clean. And we don't know if it's possible, but we have good reason to believe it is, and this is the main reason. We actually have cured some people in Hong Kong 20 years ago with terminal liver cancer. When we had good collaboration with the group there, we had 4 patients, and 3 of them were cured of liver cancer. They had nothing else to be offered. They were going to die within a few weeks. So we know it's possible, and that's what keeps us going. And by the way, there are now 30 clinical trials or so around the world with this arginine depletion. When I started on this, there wasn't really any of it in a serious way, I guess 25 years ago, not 40. And the funny thing is we think we can make asparaginase a much better drug. Whatever we learned about how arginase does the tricks and how it works in the body and so on, we think now we can take this to asparaginase and make it much more effective. If it's used in dogs as routinely as it is now, it should actually work. We think we can do that.